Okay, well, thank you for inviting me to share some of my research today on the call. I'm on the climate team, but I have a soil science background, so my project is fairly interdisciplinary. We're looking at soil ecology, the plant communities in Playa wetlands and how they're affected by climate change in the Great Plains. So I will give you some background on the wetlands that I work in, and also how climate change is impacting that region. Try to tie it back to the Missouri EPSCoR grant as well, and then give you a few of my preliminary results. I'm still in the middle of an experiment right now, and so I just have the first couple months of data ready to go. What I just said. Okay, so I work in Playa wetlands. Playas are located throughout the Great Plains, from Nebraska down to Texas. They're fairly shallow, ephemeral, so they are only seasonally inundated. They're completely fed by precipitation and runoff from the surrounding watershed. So that's why we're studying playas rather than Missouri wetlands, because the nature of the playas allows us to know that if we are changing the precipitation in our hydro period treatments, we know that that's what the wetland would be experiencing. There wouldn't be any other factors such as groundwater that would be influencing the wetland community. They're relatively small wetlands, the average size 6.3 hectares. They take up 2% of the land area on the Great Plains. So this map kind of, if you're looking at the map, I've got the outlines of the playas fairly large on here, so that's why it looks like it takes up more of the region. But they take up 2% of the landscape. And because they are essentially the only places that have water throughout the Great Plains, they serve some really critical functions. So the three that we are looking at are groundwater recharge. They're able to percolate water down to the aquifers much more quickly than the surrounding landscape. They also provide a lot of biodiversity, both in plant communities and also the animals and upper level fauna in that region. And then the third function that we're looking at is their ability to cycle chemicals running off the surrounding landscape. So most of these are going to be surrounded by agricultural row crops or pasture systems, and the playas are able to filter a lot of the nitrogen, phosphorus, and organic pollutants that are running off those fields. So I'm a soil scientist, and so the soils of the playas are really interesting to me. So the soils are mineral soils, so these wetlands aren't going to be heavily organic like those that would be in the farther north latitudes, but they do have hydric properties. And so even though they're not always saturated with water or inundated, they do have properties that show that water has been present. Another unique feature about the playa soils is that they're made of smectitic clays, which are clay minerals that shrink and swell when they're wet and dry. And so when they're dry, they develop really large cracks that can be multiple feet deep. And then when rainfall occurs, water is able to really quickly percolate down through those large cracks after the dry periods, and that's how it is able to refresh the aquifer or recharge the aquifer more quickly than the surrounding landscape. But then when they fill up with water, those clays swell and they create an impermeable layer so that it holds water. Another feature of the playa soils is that they have calcium carbonate accumulation at lower depths, and that's actually one of the ways that playas formed. Was they had these thick layers of calcium carbonate underneath them, and then as water went through and weathered the calcium carbonate, the playas subsided and created the basin in the form that they're in today. And there was also some wind involved in forming the playas. But those are just some unique features, and those features in the soils allow them to filter the nutrients because they're high clays, and so they're able to hold on to a lot of the water and nutrients that are coming into the soil. And then they also, again, with those large cracks, they're able to recharge the aquifers much more quickly than the surrounding. surrounding landscape. For the Playa hydrology, the wetlands are fairly infrequently inundated, and so they don't hold water all year round in most cases. They get typically heavy rainfall in the spring, which times well with migratory birds coming through the region, and so at that time they'll inundate usually, but then drier conditions from July to September cause them to dry out, and so by the time it reaches winter, only 1 in 11 of they're only inundated 1 out of every 11 winters is the statistic that was developed. And some of the factors that affect their inundation are the rainfall amount and the rainfall variation, which are huge driving forces for their inundation because that's how they receive water. And so it's pretty common in the Playa region because there's thunderstorms that move through that region that you could have 2 playas that are across the street from each other. One would be inundated and one would be dry based on how the rainfall moves through that region. The size of the playa affects how quickly they're inundated. Smaller playas will inundate more quickly than larger playas. The slope in the surrounding landscape affect their inundation ability, and then the surrounding vegetation. So if they're in a region that's predominantly surrounded by agriculture and row crop production where you have a lot of bare soil, you'll have more runoff coming into the playas that allows them to inundate more quickly than if you would have grassland or pasture land surrounding the playas that would catch more of the rainfall that was coming down. Like I mentioned earlier, playas are really important for biodiversity in the Great Plains. So they are able to support wetland obligate and facultative species for plants, which most of the rest of the landscape can't support. Most of those plants are going to be annuals. They develop a very robust seed bank. So typically, regardless of when they get their spring rain, they'll be able to develop some sort of plant community. So rarely would you go out in July or August and have a bare playa. At some point, regardless of how much or when the rainfall occurred, you would get something germinating in the playas. And then the species in the playas for plants are set up in zones. So in the center of the basin, there'll be more of the wetland obligate species. And then as you move out towards the rim, there's more moist soil plants like smartweeds and barnyard grass. And then as you move, as you're transitioning out into the uplands, then you'll start to get more upland plants. And so we're interested in seeing if we change the hydro period in the future and you don't have as much water coming into these playas, if you'll get those upland plants encroaching on the center of the playas, which could have an effect on the wetland-dependent species that use the playas. And then lastly, so plant biodiversity is important, but the playas also are supporting migrating water birds and other wetland-dependent species like invertebrate communities, amphibians, reptiles in that region as well. I'm not a climate scientist. I'm going to try to talk about climate change as well as I can, but in the Playa region, we know that the temperature is expected to increase, and the graphic I'm showing here on the screen is from the CMIP 3 IPCC third assessment, I believe, and it shows that in the Playa region, there's supposed to be a 4 to 9 degree Fahrenheit increase in temperature by the end of the century, and throughout the region, on this map, it shows that Nebraska and Texas are going to be affected similarly. From the northern end to the southern end of the Playa region will have similar temperature effects, but I've seen other models where Nebraska might be more heavily affected by temperature increases than Texas or vice versa. For precipitation, there's a lot of uncertainty, but they are expecting that there's going to be more consecutive dry days in these regions, so less ability for the playas to be inundated, and then also an increase in heavy rainfall events. So climate change has been studied and how it affects wetlands. Primarily, we know that in the northern latitudes, there's likely going to be a reduced number of wetlands due to warming because they won't be able to support as much or hold as much water. When wetlands experience climate change or warming, they tend to allocate carbon into below ground biomass. So that might be a shift that we see in the plant communities in Playa wetlands. And then we know also that humans have reduced the ability of Playa ecosystems to respond to climate change, and that's primarily due to sedimentation, which I'll talk about in the next slide. And some of the most pressing impacts that have been identified for climate change in wetlands are their ability to maintain habitat for wetland-dependent species, how it's going to shift the greenhouse gas emissions coming from the wetlands, and then also the flood control, which we aren't as much interested in the Playa region, but that's going to be something that might affect wetlands in other regions with climate change. Land use change is something that's been fairly well studied in the Playa region. So because of the surrounding row crop agriculture, a lot of sediment runs into the Playas, and that has caused the Playas to hold less water, and it's decreased the biodiversity in the Playas. Predictions show that the Great Plains are going to face a continuous loss of natural resource land. However, studies have also shown that when environmentally oriented policy is put in place, that it's able to conserve natural resource land in Playas. And so while these changes are happening, there's also some opportunities for policy and management techniques to be put into place that would help conserve the natural resources in that area when we start facing climate change and land use change. So with all that in mind, so the objectives for my project are just to better understand the changes that are occurring in Playa wetland plant species, and the soil chemical properties that are associated with predicted changes in precipitation and temperature that may occur with climate change. So there's just not been a lot of studies done in the Playa region and how those wetlands are affected by climate change, and so we're trying to understand some of the specific processes that are occurring that might be sensitive to changes in precipitation and temperature in the future. I've got 4 sub-objectives to my dissertation project. I'm only going to talk about 2 today because those are the ones that I'm looking at in my first 2 experiments. So the first is to quantify the effects of simulated climate change conditions on the following properties. Sorry, I shouldn't read off my slide. But we're looking at the plant germination, growth, seed biomass, and the species diversity in Playa soils. Also looking at soil chemistry and biogeochemical processes in the soil solution and in the soil, the bulk soil itself. And then I'll be following it up with an experiment to look at the greenhouse gas emissions coming from Playa soils, and we're also trying to get funding to look at the microbial community dynamics as well. And specifically in this project, we're looking at the hydro period changes and precipitation changes rather than temperature. And then the second thing that we're trying to do is look at differences that are occurring between the northern and southern Playas, so between Nebraska and Texas, where I've collected my samples, just because a lot of studies have been focused on one region or the other and haven't done a lot of work with seeing how those 2 regions are going to be affected differently. So for this experiment, I collected soil from 10 Playas, 5 in the rainwater basin region of Nebraska and 5 in the southern high plains region of Texas. So I collected those last spring before germination occurred. My Playas were selected based on the conditions at the time. I'm sampling around the rims of Playas in the moist soil region where the plant communities are potentially going to be most vulnerable to encroachment from upland plants. And so when I went out to collect my soil cores, it would have been really difficult to collect them if the wetland was completely inundated. And so I had a plan, and I went to a couple. They were full of water, and I couldn't sample them at the time. So I worked with experts in the region to adjust my sampling plan. But they all had pretty typical landscape features. In Nebraska, they were mostly surrounded by row crop production. In Texas, most of my Playas were surrounded by pasture land that were grazed with cattle. And then we took samples as well to do initial soil characterization to make sure that there wasn't too much variability, or basically so we could understand the variability that was occurring between our sites. For the climate information, our historical climate data, we used NOAA weather stations that were located in Hastings, Nebraska, and Lubbock, Texas, which were the centers of the two regions where I sampled. And we looked at the last 30 years of data to get averages for our historical data. For our future climate scenarios, we used CMIT-5, BCCA downscaled atmosphere-ocean general circulation models for RCP 4.5. And we calibrated with the last 30 years of data, and then we are looking at end-of-century climate simulation, so from 2070 to 2099. Initially, we ran 19 models. We adjusted those models for excess drizzle days using a procedure that Dr. Savoma has worked on with some students in ag engineering. And then we also used quantile mapping to capture the extreme events that were going to occur. And for this first experiment, we were primarily interested in the daily precipitation in millimeters per day, so that's how all these were examined. From the 19 models that we ran initially, we selected 3 to use as our climate treatments. So the first one was a wet scenario, and that essentially was the model that had the fewest dry days and the highest amount of precipitation compared to all the models. And the one that was selected was the Community Earth System model, coupled with biogeochemical parameters from the National Center for Atmospheric Research in Boulder, Colorado. The dry scenario was the opposite, the one with the most monthly dry days and the lowest monthly precipitation compared to the rest of the models. And for that one, we selected the Max Planck Institute Earth System model based in Germany, or made in Germany. And then the third scenario that we selected was just an average of all of the models. And so that was the one with the lowest deviation from model averages for monthly dry days and monthly precipitation. And that one was ended up being the Muroc Earth System model coupled with chemical factors that's produced in Japan. We also, so when I do my greenhouse experiment, I wanted to be able to apply a precipitation amount each day in millimeters per day onto my experimental units. And so to do that, rather than averaging all the years from that model, we selected a year that was the most similar, or that represented the average of that model over the 30-year period we were looking at. And so that way it would give us 1 year where we would have data from April to October to say on April 1st, the rainfall amount is going to be this many millimeters. On April 2nd, it's going to be this many millimeters. And that's allowed us to have consistent treatments for our hydro period study. And over here on the slide, you can see the rainfall amounts over the growing season that we developed from these models. So Nebraska, you can see, has more rainfall over the growing season than the Texas samples, and the historic treatments are close to our future wet treatments for both Nebraska and Texas. The future average and future dry scenarios are going to see much less precipitation over the growing season than the historical numbers based on these models. Okay, so getting to my experiment. So I am currently in the process of completing a 6-month greenhouse experiment. So we collected intact cores from the field, like I mentioned earlier, and if you're able to see the presentation, you can see how they're set up in the greenhouse. So they're randomly distributed around the greenhouse. We are using the natural seed bank coming up from these playas. So we collected them before germination occurred, so that way we wouldn't have germination until we started applying treatment. That means that we have a very diverse plant community, which I'll talk about when I get to my results. But it also means that we have a lot of site variation. And so I'm including site as a random effect in all of my analysis just because there's a lot of variation between the playas where we collected our samples. To start the experiment, we had ideal conditions for moist soil germination. So we did that for 28 days. We held them at constant moisture to allow for plants to start germinating, and after that we started to apply our precipitation treatments. We have 7 treatments, so we have 10 replicated playas, and then each one has 7 treatments. So we have 3 control moistures, one that's a moist condition, one that's saturated, and one that's at a dry wilting point condition. And then our 4 climate scenarios are the historic climate conditions, future average climate conditions, future wet climate conditions, and then future dry climate conditions based on the climate models that we selected. We're trying to look at how everything is interacting within the ecosystem, and so we are collecting quite a bit of samples. So we collect plant samples every other week. I count the number of plants and record their height and their growth stage. Also, we are collecting SPAD content information to look at relate that to the chlorophyll content in the leaves. We're looking at the species composition, so the diversity amongst the plant communities. And then at the end of the experiment, we'll collect above and below ground biomass information and also collect seed biomass. And the seeds have already started dehiscing, so I've been collecting those for a while. But hopefully we can see some variability in how the biomass is being allocated in different scenarios. We're also collecting in situ conditions, so we have soil temperature and redox potential probes in one of each treatment from the 2 regions. So we have 7 of these in Nebraska, 7 in Texas samples, and that allows us to get a snapshot of what's going on in the soil continuously as we add the precipitation treatments. And then I collect soil samples monthly to measure the soil biogeochemical properties, and then also we will hopefully be using those soil samples to look at the microbial metagenomic community as well. And then the soil solution sampling, we were hoping to do weekly, but we've had some troubles getting our lysimeters to collect water. It turns out that if there's no water in the soil, it's really hard to pull water from the soil. And so we've had a bit of trouble getting consistent measurements on those. So I won't be talking about any of that data at this time. So for the preliminary results, we did our initial soil characterization at the Soil Health Assessment Center on campus, or over at South Farm, I guess. But the physical properties are pretty much exactly what we would expect for playas. We found pretty heavy clay textures. Texas had much more clay than Nebraska, but both of the regions exhibited characteristics that we would expect from a typical playa. And then the chemical properties as well are pretty typical of what we would have expected from the playas, and they're also fairly similar. The pH and electrical conductivity are similar to what we're getting when we take our solution samples in the greenhouse as well. So we've got those redox potential probes that are in the soil and are able to respond immediately when we have precipitation events. And so redox potential is something that shows us how likely it is that the microorganisms in the soil are going to use an electron acceptor other than oxygen. So aerobic organisms prefer to use oxygen as their terminal electron acceptor when they are metabolizing carbon that's in the soil, and that's how they get their energy. So when oxygen is absent, they will move towards using other chemical species. So one of the important transitions that happens once you start getting low oxygen conditions is that nitrates, NO3- will start to be metabolized or transition into nitrous oxide or dinitrogen gas. So both of those then would be emitted or volatilized from the soil. And that's one way that these wetlands are able to reduce the amount of nitrate and processed nitrate that's coming into the soil from the surrounding landscape. So on this chart here, there's a lot going on, but the diamonds show the precipitation that's occurring, and then the dotted line is the redox potential. So a lower redox potential means that there's less oxygen in the soil. And then there's the line there that says anoxic border, and that's the anoxic border that shows when nitrate is going to be reduced to produce dinitrogen gas. And this is pH dependent, so this is at pH 6.5, which is fairly typical in our playas. But you can see that at the beginning in the spring, we had conditions that were causing the redox potential to get below that anoxic border. So we would have had nitrate being used as a terminal electron receptor or acceptor at that point. But then when we get later in the season and we have lower precipitation amounts of precipitation, then oftentimes our redox potential isn't going to be pushed low enough where we start to see that transition occurring. So this graph then is kind of messy. This is trying, so that last graph is showing for the wet treatment in Nebraska playas, and this is a graph that shows all of the Nebraska and Texas climate treatments. So the orange color is all the Nebraska treatments, the blue color is the Texas treatments, and you can see that it's fairly similar. At the beginning of the season, we had enough rainfall that it was pushing the redox conditions below that anoxic border, but then as we start to get less precipitation in the summer, the redox potential starts to increase and no longer reaches that anoxic border unless there's pretty extreme rainfall events. And one thing that I found was interesting is that the Texas playas actually maintain the lower redox potential for longer periods of time than the Nebraska playas after rainfall events. And that's probably because they have more clay, and so the water doesn't evaporate as quickly. My plate communities, like I said earlier, are incredibly variable in between the different treatments and the different sites. So you can see the picture on here. I had a lot of growing plants. Now they're all dead, so they're not very pretty anymore. But I do have some that are still persisting. I have a good mix of broadleaf and grass plants. One thing that I can kind of see in some of my samples is that as you get into more dry conditions, you start to have more broadleafs than graminoids, and so that might be something that we can look at in the future. It wasn't something that we were intending to look at, but the life histories and the types of plants might be more affected by the climate change conditions than just looking at diversity index. And so this is just a preliminary graph of the diversity index values that we've calculated, and what we're finding is that at the beginning of the study we had more diversity in our historic climate treatment, and then as we've moved throughout the experiment, and I think this is probably even more of an effect now, but I don't go all the way until our current results, but the dry climate treatment actually becomes the most diverse of the treatments. So the dry future conditions in both Nebraska and Texas tend to develop more diversity, and I think it's because their precipitation values are lower, but they also get more intermittent rainfall events, like small rainfall events throughout the growing period, versus the other three treatments, the historic climate, the future average, and the future wet. Those are getting more extreme rainfall events in larger amounts when they get rainfall, but that future dry that's getting just a little bit of rainfall every now and then is actually showing us more diversity in the species. But like I say on the slide here, there's no significant effect of treatment so far. mostly we get an effect when we incorporate the site as a random effect, and so we get an interaction effect between treatment and site. Those are all the results I have to share right now. I am in the process of getting a lot of the soil samples processed, and then also figuring out how we're going to look at the solution samples since they're pretty sparse. But we're hoping to, by the end of this experiment, be able to identify the processes that are happening within playas that are most sensitive to the precipitation changes that we're subjecting onto them. And then in combination with the other experiments that I'm going to be doing, a germination experiment, one to look at the greenhouse gas emissions, and then also one to look at if we increase nitrogen coming into the playas, how the community responds. But in combination of all those experiments, we're hoping to develop playa vulnerability indices that will give managers and landowners an idea of which playas are going to be sensitive to future climate conditions. And that will hopefully assist in managing the playas and helping landowners and managers in the playa region to conserve the playas and make sure that they're still able to function as biodiversity units on the landscape, and also recharge the aquifers and process the nutrients that are coming off of the surrounding landscape. So just a quick update on some other things going on with this project. We just applied for a grant to help us fund some of the microbial metagenomic analysis that we want to do with this project. So in our EPSCoR funds, we don't have quite enough to look at the microbial populations in the soil, but those are really important in driving a lot of these processes, and so we'd like to be able to look at how those communities shift with the climate change. I'll also be presenting this research at the Soil Science Society of America meetings next week in Phoenix, and at the USGS Missouri Cooperative Fish and Wildlife Unit meeting here next week. One of the fun things about this project is that my co-advisor, Lisa Webb, is in the USGS co-op here, and so it's been fun working with that group and learning more about the ecological aspects of the project. And then in February, I was invited to give 2 talks, primarily because I'm working in playas, but the Rainwater Basin Joint Venture, which is located in South Central Nebraska, I've been invited to talk at their informational seminar, which is primarily an audience of landowners and land managers, and then also at the Midwest Fish and Wildlife Conference and a Playa Symposium in February. So the EPSCoR information is going to be getting out. And with that, I'll take any questions. I don't know where I'm at on time, but.
Thanks, Rachel. This is Bo. I think that you did a really good job in figuring out how to pull out the information you wanted from all those climate model time series. And I was just curious on the details of how you chose these specific years as a treatment, the specific single years to use.
Sure. So I looked at everything in monthly averages, and so for every month in every year of the model between April and October, I calculated the precipitation total for that month and also the total number of dry days in that month, so the days without any precipitation. And then I took an average of all those years, and then I looked at, I chose the model where it had the lowest deviation in dry days and precipitation, if that makes sense. And so it was hard to actually capture both of those things, I guess, but I chose the year where, like, quantitatively there was the lowest deviation from the model average in precipitation, and then the lowest deviation, like just like total number, like that deviates from the model average of total dry days. Does that make sense?
Yeah, that's what I figured. Thank you.
Okay.
Hi, Richard. This is Sun Song from UMPC. It's an excellent presentation. My question is, you look at climate change impact on the wetlands of Playa. Do you have any comments about the feedback? Let's see the shrinking of the area of Playa. What's the feedback to the climate change, especially in a regional scale?
Okay, so let me repeat this, make sure I'm understanding. You're asking about what feedback the climate change might have on the wetlands?
No,
so if there's
yeah, the feedback from the wetlands on the climate, on the regional climate.
Oh, okay. So I guess one of the bigger things that is talked about with wetlands and climate change is the greenhouse gases that are being emitted. I don't know if this is a proper way to answer this question, but I think it's interesting. So playas are not going to be typically wetlands that are producing methane. Methane requires long inundated periods of time, and most playas are quite ephemeral and don't hold water long enough to be producing methane or hydrogen sulfide. However, if they did stay wet longer, then you would see more of those gases being emitted, so more of those volatile compounds. And then also with warmer temperatures, then you're able to have microbes working harder and are going to produce more gases because activity increases as temperature increases. So that's I guess what I've thought about before. I haven't thought about the hydrology necessarily, and if there's any feedbacks that go along with that.
Yep. Great. Thanks.
Rachel, I
Okay.
have a question, but I may have missed something because I joined late. So this is just anecdotal, but observation when I was in West Texas recently, looking at some dried up playas, it looked like the vegetation within the playa area was like very noticeably different from the surrounding area. So in general, is that the case? And is there like quantitative information about like the species distribution within the area that's actually part flooded and then dries?
Yes. So the playas do support a much different plant community than the surrounding landscape, and so usually in the center of the playas is where you're going to get like obligate wetland species because it stays wet usually long enough in the spring that they're able to germinate. In the playas in Nebraska, you see this a little more, so like you'll have more emergent vegetation in the centers, like cattails, bulrush, and then as you move out in the playas, you'll move to a zone that has moist soil plants like smartweeds, polygonums, or that's the same thing, sorry, and then grasses like barnyard grass, millet. And then as you move towards the far outside, then you'll start having more upland species of plants. And I attempted to sample from the moist soil region, and so my plants in the greenhouse are mostly exhibiting characteristics of those that would grow on the rim of a playa. So the moist soil plants like smartweed and barnyard grass. I have lots of smartweed and barnyard grass in my playas. Does that answer your question?
Yeah. I mean, it seems, just visually, I didn't get close enough to take a careful look at the different plants, but it seemed pretty obvious and interesting from an ecological perspective.
Yeah, it was funny. I had to go take my samples in the spring, and I was really concerned about being able to identify the moist soil plant zones while all the plants were dead and not growing. And it was so obvious where I needed to take my samples because the polygonum is that red color on the landscape. And so it was really easy to identify the moist soil region even when the plants aren't alive.
Cool. Thanks. Any more questions for Rachel? No? Going once? Okay, thanks, Rachel. That was great. A nice intersection between climate and plants, which is just what we want.